Our top answers are currently up on the board. We asked 100 College of Science students, name the top generalized concepts to understand the Ebola virus.
Well, I know Ebola is a single-stranded RNA virus that infects both humans and primates, so the answer is going to be virus that causes disease.
You know, Steve, I think one of the first generalized concepts they should know would be the first outbreak.
Well, everybody knows that Ebola is spread through contact with bodily fluids and not through the air, so I'm going to go with transmission. I think people are going to know about the mortality rate of Ebola. It's got a 50% death rate, and that's due to once you get the virus, it causes issues with your blood clotting, and you die due to organ failure and fluid loss. You know, I bet that last one up there is vaccine development. I know that vaccines got developed and they will protect you for an entire life after just a single dose that you can get started at just 12 years old.
The Ebola virus disease is a human-to-human transmission through direct or indirect contact with blood or bodily fluids. This also includes secretions from infected patients. Ebola virus cannot be transmitted through the air. With Ebola having such a high mortality rate, you may be wondering how to manage outbreaks. The first and most important thing will be community engagement as well as patient education. This also includes case management, infection prevention, surveillance, and contact tracing. Vaccines have also been developed to help protect against outbreaks, as well as 2 monoclonal antibodies were approved for treatment infection by the FDA in 2020.
We asked 100 immunology students, what are aspects of innate immunology in reference to the Ebola virus?
One of the answers that will be up there is definitely going to be PAMPs. They activate PRRs on the TLR4 pathway.
Well, I know that the Ebola virus affects innate immunity by activating the lectin pathway of the complement system, so I'm sure that's up there. Those same innate immune system cells release IL-1 beta, IL-6, and TNF to fight off the Ebola virus.
One of the things that innate immunity is going to be affected by with the Ebola virus is definitely going to be a delayed type 1 IFN response in bystander cells.
Right over here, Steve. I know that the innate immune system is activated as soon as the Ebola virus enters the organism through the mucous membrane. Steve, I know that the Ebola virus affects macrophages, monocytes, and dendritic cells, which all make up the innate immune system. Well done, Team Haley. Now let's take a deeper dive into the innate immune system. Evaders invade the organism through the mucous membrane and lead to effects on the macrophages and dendritic cells. Macrophages view the PAMPs through TL4 and release transcription factor NF-kappa B, which releases TNF, IL-6, and IL-1 beta, which can lead to a cytokine storm, which is not good for the organism. This leads to the activation and degranulation of neutrophils to fight the infection and killing Ebola-infected cells. These cytokines in response lead to whole body inflammation. Dendritic cells affected by Ebola have suppressed ability to produce interferons and cannot produce IFN alpha or IFN beta, allowing for Ebola to spread through the organism. To help destroy Ebola-affected cells, the complement system is activated through the lectin pathway, which is when MBL binds the pathogen membrane, activation of C4 and C2, formation of the C3 convertase, formation of the C5 convertase, leading to the membrane attack complex and destruction of the infected cell. The repercussions of Ebola on the organism lead to organ failure and hemorrhage.
We asked our lecture PowerPoints, what are aspects of B cells in connection to the Ebola virus?
This is a great question. So when the virus enters the body, there's actually a complex that gets made that includes the virus, antibodies, and a C1q complex. This actually promotes the binding of the virus to Ebola virus receptors to enhance that binding.
Well, the Ebola virus is going to affect the B cells in the adaptive immune system by increasing the level of cytokines of CXCL13, which is a known biomarker in germinal centers, which lead to the production of B cells.
Generally with B cells, after infection is happening, the cell will go under lots of rounds of migration as well as proliferation. So in relation to the Ebola virus, there's definitely going to be some affinity maturation and isotype switching as well as that proliferation and migration. The general process of B cell activation includes the B cells being moved to the secondary lymphoid tissue. This will be coming in through the high endothelial venules. This will then produce cognate pairs and include proliferation in the cell. Inside the B cell zone, those proliferated cells will move to the T cell zone and then secrete antibodies. These antibodies will be included in IgG and exit through the efferent vesicle. How does Ebola affect B cells? Ebola will increase the levels of CXCL13, which is a cytokine, also known as a signaling protein. CXCL13 is a biomarker that represents germinal center activity. When a germinal cell is activated, what does that mean? When there's an increase in germinal activity, this will increase proliferation, it will increase immune response, but also decrease self-tolerance, meaning there will be a heightened immune system reaction. Now that you know the effects of the Ebola virus, we will now see the complex that is formed from the infection. It will include the virus, antibody, and C1q complex that will produce the increased proliferation and immune response.
We asked the Cougar nerds, what are the importance of T cells in regards to the Ebola virus?
Ebola virus affects the T cell aspect of immunity due to the fact that it builds both CD4 and CD8 T cells, but there's a stronger response for those CD8 cells.
Infected dendritic cells actually have an impaired ability to activate analogous T cells. This means that activation is actually a key part of the T cell's life cycle that is affected by Ebola.
Steve, I know that MHC class 2 has antiviral properties that work with T cells to fight against the Ebola virus once they have activated the immune system.
General information about T cell immunology begins with a dendritic cell being activated. This will be activated through PAMP signaling. Dendritic cells express cell surface receptors all around itself, and then that dendritic cell will then migrate into the secondary lymphoid. Dendritic cells express antigens on MHC molecules. T cells will then bind to MHC molecules, and then CD8 will then bind to B7 on the dendritic cell surface. T cells will release IL-1 beta and bind with receptors on other T cells, or they will bind with the same T cell receptors on its own cell. When it comes to T cell immunology and its effects from Ebola, Ebola will actually infect the cell surface, making that cell an infected cell. An infected cell will only produce dendritic cells that are damaged, and with damaged dendritic cells, T cells will not be activated. This will limit the capabilities of producing a protection against the virus. Specifically, MHC class 2 capabilities on the surface of dendritic cells will actually have strong antiviral capabilities against Ebola virus. Without that T cell binding, the immune system will eventually have led to an overwhelming viral load by Ebola.